Hello, can you hear me?
Yes.
Okay, we can see you.
Mhm.
Everything okay?
Should we start now or later?
Yes, please start the lecture.
Okay. Hi guys. I can't see anyone, but I know you're there. My name is Wu Di. I'm from Wenzhou Medical University School of Pharmaceutical Sciences. Working in the same group with Professor Wang and Professor Liang. We were from the Chemical Biology Research Center. And actually, I have some connections with BSMU because some of your professors visited our university, and our colleague also visited yours. So I was a host when your professors come to visit Wenzhou Medical University. So I'm very glad here to give you a brief lecture. Okay. Can you see my PowerPoint? Okay, I think you can. Well, my topic is the coming age of post-defense peptide. And actually, it is my research area, so I can give you an introduction about post-defense peptide and how we do research on the post-defense peptide. I'm going to introduce from these 5 aspects. What is host defense peptide and the function of host defense peptide, and basic knowledge about amino acids and peptides and secondary structures. And I will show you one of my study, a case. And later, if we have enough time, I can give an introduction about peptide bioinformatics. So let's answer the first question. What is host defense peptide? And HDP is short for host defense peptide. We also call this HDP. Host defense is also known as antimicrobial peptides traditionally because you can see the name antimicrobial. So the function may be about the anti-microbes. And they are actually ribosomally synthesized and relatively small, less than 10 K-Dalton, and usually cationic, amphipathic, and of variable length, sequence, and structure. The classification of host defense peptides are based on circular dichroism studies, CD studies. And many of them have been supported by NMR, nuclear magnetic resonance, and include about 5 major classes: alpha helical structures, beta sheet structures, and loop structures, and extended structures, and some of them are cyclic structures. And the host defense peptides, they are found by all multicellular organisms. Like I said, the name is antimicrobial peptide. Well, later I may talk about the HDP or AMP antimicrobial peptides, but you know they are the same thing. So the antimicrobial peptide, they can kill bacteria and yeast, fungi, viruses, and even cancer cells. So and that's why we don't call it antimicrobial peptides anymore. Because there are many more functions other than the antimicrobial function. And now they also find a wide variety of species ranging from bacteria to insects to humans, and they can be expressed or induced in response to infections and inflammations and cytokines. Well, the host defense peptides are produced by all complex multicellular organisms, like I just said, bacteria, animals, even human bodies. But we can show you from about the history of the discovery of host defense peptide. Well, the peptides discovery likely started in the 1960s of Bombini in frogs and lactoferrin from milk. That's 1960s. And in 1980s, a landmark study was studied by Hans Bowman, and he reported how injection of bacteria into the Cycropia silk moth and induced a potent antimicrobial peptide. And this discovery of cycropins, this peptide is called cycropins, was the first major report of alpha helical antimicrobial peptide. And another important advance was from Zeslov and his colleagues. Zeslov was very famous in this antimicrobial peptide area. And the peptide was isolated and characterized from African cloud frog, and it's called maganin. And in the 1990s, Hoffman's group showed that genetic ablation of antimicrobial peptide synthesis rendered the fruit fly susceptible to a massive fungal infection. And this provides the first evidence of the critical role of antimicrobial peptides in insect host defense. And the discovery of antimicrobial peptides in mammalian skin and the subsequent generation of mice deficient in one of these host defense peptide clearly demonstrated that the relevance of antimicrobial peptides and to mammalian host defense system. So antimicrobial peptides have since been extensively characterized and discovered in virtually and all multicellular organisms that have been studied for this activity. You must know Alexander Fleming from his achievement on the discovery of penicillin, but actually he has another very important discovery that is lysozyme. It is the first reported human antimicrobial protein because it's a little bit large, so it's a antimicrobial protein. It was identified in 1922 from nasal mucus by Fleming. And this observation was overshadowed when in 1928, Fleming discovered penicillin. So in the 1940s, he and along with others, Chen and another guy, they were awarded the Nobel Prize, right, for medicine. That's the beginning of golden age of antibiotics in the 1940s, led to a loss of interest in the therapeutic potential of natural antimicrobial protein or peptides, such as what he has had discovered lysozyme. But in the 1960s, the rise of multi-drug resistant microbial pathogens awakened the interest in antimicrobial peptides as host defense molecules. So now we mentioned the multi-drug resistant microbials. Let's see what is drug resistant and what is multi-drug resistant microbials. Let's see this short video.
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total biomass is greater than that of all plants and animals combined. And they live virtually everywhere, on the ground, in the water, on your kitchen table, on your skin, even inside you. Don't reach for the panic button just yet. Although you have 10 times more bacterial cells inside you than your body has human cells, many of these bacteria are harmless or even beneficial, helping digestion and immunity. But there are a few bad apples that can cause harmful infections, from minor inconveniences to deadly epidemics. Fortunately, there are amazing medicines designed to fight bacterial infections. Synthesized from chemicals or occurring naturally in things like mold, these antibiotics kill or neutralize bacteria by interrupting cell wall synthesis or interfering with vital processes like protein synthesis, all while leaving human cells unharmed. The deployment of antibiotics over the course of the 20th century has rendered many previously dangerous diseases easily treatable. But today, more and more of our antibiotics are becoming less effective. Did something go wrong to make them stop working? The problem is not with the antibiotics, but the bacteria they were made to fight. And the reason lies in Darwin's theory of natural selection. Just like any other organisms, individual bacteria can undergo random mutations. Many of these mutations are harmful or useless, but every now and then, one comes along that gives its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives quite the edge. As the non-resistant bacteria are killed off, which happens especially quickly in antibiotic-rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that help them do so. Reproduction isn't the only way to do this. Some can release their DNA upon death to be picked up by other bacteria, while others use a method called conjugation, connecting through pili to share their genes. Over time, the resistant genes proliferate, creating entire strains of resistant superbacteria. So how much time do we have before these superbugs take over? Well, in some bacteria, it's already happened. For instance, some strands of Staphylococcus aureus, which causes everything from skin infections to pneumonia and sepsis, have developed into MRSA, becoming resistant to beta-lactam antibiotics like penicillin, methicillin, and oxacillin. Thanks to a gene that replaces the protein beta-lactams normally target and bind to, MRSA can keep making its cell walls unimpeded. Other superbacteria, like Salmonella, even sometimes produce enzymes like beta-lactamase that break down antibiotic attackers before they can do any damage. And E. coli, a diverse group of bacteria that contains strains that cause diarrhea and kidney failure, can prevent the function of antibiotics like quinolones by actively booting any invaders that manage to enter the cell. But there is good news. Scientists are working to stay one step ahead of the bacteria, and although development of new antibiotics has slowed in recent years, the World Health Organization has made it a priority to develop novel treatments. Other scientists are investigating alternate solutions, such as phage therapy or using vaccines to prevent infections. Most importantly, curbing the excessive and unnecessary use of antibiotics, such as for minor infections that can resolve on their own, as well as changing medical practice to prevent hospital infections, can have a major impact by keeping more non-resistant bacteria alive as competition for resistant strains. In the war against superbacteria, de-escalation may sometimes work better than an evolutionary arms race.
Okay, that's a short video about the super bacteria. It's also called the multi-drug resistant bacteria. They are also called superbugs. So we know the antibiotics were capable of treating most bacterial infections, but the emergence of these superbugs diminished the effectiveness of existing antibiotics. So we can see why we call the host defense peptide the coming age. Let's see this graph. Well, the increased frequency in the number of isolated drug-resistant pathogenic microbes in hospitals worldwide has promoted both research institutions and pharmaceutical industries to design and develop novel drugs targeted against resistant pathogens. And interest has focused on host defense peptide, primarily due to their broad spectrum activity and low potential to induce resistance and diverse range of activities. Well, recently studies have demonstrated a wide range of activity in modulating the function of host cell and tissues as part of natural host defenses, which has led the depiction of this peptide as host defense peptides rather than, like I said, the antimicrobial peptide to better define their multi-phase as immunomodulatory mediators. And also under some circumstances, they are antimicrobial agents. Well, here on the right, you can see on the red section indicated by the dashed yellow lines. That's the resistance mechanism in gram-negative bacteria, like transmembrane efflux pump or big lactamases. It's all antibiotic inactivation. And on the left section is indicated by the dashed yellow lines of the bacterial cell, shows the mechanism of action of these host defense peptides. And here is the more detailed mode of action. Generally, antimicrobial peptides or host defense peptide exhibit a net positive charge and a high ratio of hydrophobic amino acid, which allows them to selectively bind to the negatively charged bacterial membrane. The net charge of human cell membrane is always neutral, but for the bacterial cell wall and cell membrane, it's always negatively charged. So there is a positive and negative charge action. And the polyanionic, the negatively charged microbial cell surface contain acidic polymers such as LPS lipopolysaccharides, and wall-associated teichoic acid, TA, in the gram-negative and the gram-positive bacteria, respectively. They transit the outer membrane of the former while self-promoted uptake. And selectivity for the specific species is due to the differences in the membrane composition of different microbiomes and cell types. And subsequently, these peptides contact the anionic surface of the cytoplasmic membrane and insert in a manner such that they initially straddle in the interface of the hydrophilic height group and the fatty acyl chains of the membrane phospholipids. And after insertion into the membrane, antimicrobial peptides act by either disrupting the physical integrity of the bilayer while membrane thinning, or transient proliferation and or disruption of the barrier function or translocate across the membrane and act on the internal targets. Some complex and conventional models already describe these subsequent events, including the reorientation of peptide molecules perpendicularly inserted into the membrane to form as a barostave or toroidal channels to break down the membrane integrity, and as a result of the swamping of the interface, the detergent-like dissolution of the patches of membrane and the formation of membrane charged by a carpet of peptide at peptide lipid aggregates within the bilayer. And each of these successfully predicts the ability of cationic peptides, antimicropeptides, to break down the cytoplasmic membrane. That's generally the mode of action of antimicropeptides. It's mostly working on the cell membrane and the cell walls. But still, they can modulate host immunity. Modulating the immune response and regulating inflammations. Like here, the peptides and host immune factors is on the right panel. The antimicropeptides can act as chemoattractants that initiate the recruitment of innate immune cells to the infection site and initiate the chemokines induction by various cells. Well, in some chronic autoimmune diseases, the cytokine-induced cathelicidins, cathelicidins is antimicropeptides from the human body. They can induce the cathelicidin expression and overexpression of human defenses, confers protection against skin infection, whereas the anti-inflammatory nature of the peptides can suppress both chronic and acute inflammatory conditions by influencing the toll-like receptors, TLR, and toll-like receptors. This signaling pathway is also a main research area in our group. So by influencing the toll-like receptor recognition of microbial products and the nucleic acid released upon the tissue damage. Here is the complex response of mouse macrophage to human beta defensin 3. Beta defensin is also antimicrobial peptides from human body, HBD3. And left panel is a network interaction diagram that shows proteins belonging to the interleukin signaling pathway, the blue ones, and the lipid and the lipoprotein metabolism pathway, the red ones. And these are a subset of the gene that changed expression after treatment with human beta defensin 3. As determined using softwares. Although these pathways accomplish divergent functions within the cell, they are interconnected by share several common proteins. We can see here the purple ones. And for the right panel, it's a summary of the expression patterns of cytokine signaling genes in macrophages activated by endotoxin, which is RPS lipopolysaccharide, the endotoxin, alone or endotoxin together with human beta defensin 3. Although cytokine signaling is thought to be dampened by the presence of the cathelicidin LL37, a very important host-defense peptide in human body. And many genes belonging to this pathway are upregulated by human beta-defensin 3. And the network diagram here is generated using our software network analyst 8. We can show you more about the human body, human defense peptide. Well, for human body, diversity of host defense peptide activities within the body and the relationships are showing here, and they are also always with the associated with the disease states. Host defense peptides are produced by various different cell types throughout the body. Here are HDPs that are produced by the epithelial cells of the skin, lungs, and the gut, as well as the immune cells of the circulatory system. On the skin, certain human host defense peptides, such as the cathelicidin LL37 and human beta-defensin, the HBDs, and human neutrophil peptides, HNPs, they are constitutively expressed by keratinocyte and mast cell, whereas the expression of others can be strongly induced in response to injury or infection, which attracts immune cells to this area surrounding the damaged tissue. And in the lungs, host defense peptides produced by airway epithelial cells help to protect from invading microorganisms. And in the gut, the penis cells produce large amount of human alpha defensin 5, HD5, and HD6 at the base of intestinal crypts, helping to prevent the infection from pathogenic bacteria and maintain homeostasis of the commensal microorganism community. In the circulatory system, many immune cell types express HDPs in response to infection and inflammation. These peptides can have multiple effects throughout the body, depending on the location where they are produced and the type of cells that they present. Dysregulation of host defense peptide expression at any of these sites can contribute to various disease states. Well, that's a basic introduction about host defense peptide. So let's think about this. What has affected these different functions? You can see on the red panel, here we have mentioned about alpha defensin and human beta defensin 3, HBD3, and LL37, the human cathelicidin, because there are 37 amino acid residues, so it's called LL37. And from bovine, the indolecidin and niacin, polymyxin B is a non-ribosomal antimicrobial peptides, but now it's on the market, polymyxin. So we can see the differences between these host defense peptides. That's their structures. Yes, that's their structures affected their different functions. So let's go to have a basic study about the basic structure of these peptides. Let's start from the amino acid and the peptide biosynthesis. Here is the, you must have studied this in the biochemistry and molecular biology, I think. So let's recall the eukaryotes, the thing, we call that the central dogma of molecular biology, right? Here is the DNA in the nucleus, and each strand of the DNA holds a code to create specific proteins because the DNA can't leave the nucleus of the cell, and a copy of the code called messenger RNA, mRNA, is made, and this is called transcription. And then the mRNA takes this information outside of the nucleus and bring it to the ribosome. And the ribosome move along the mRNA reading the code, and this is called translation. Now we have transcript and then translated. Another type of RNA called transfer RNA, tRNA, collects the specific amino acids that are needed to make the protein. And then the tRNA brings the amino acids to the ribosome, and the ribosome then build a chain of amino acids in the proper sequence based on the code in the mRNA called elongation. And then the ribosome contributes to move down the mRNA strand until all the appropriate amino acids are added and the protein or peptide is complete. So we can see the basic blocks of the peptide chain or protein chains are amino acid. Here is a structure of amino acid. Why we call it amino acid? Let's see the structure. Here is the central carbon, the alpha carbon, because this is the functional group. So the carbon besides the functional group is called alpha carbon. The alpha carbon connected to, they have 4 bonds, and one is acid group. The other one is amine group. So it's called amino acid. And there is another hydrogen here connected to the alpha carbon. And there is a side chain also connected here to the carbon. And we always call it alpha amino acid because the amine group is here connected to the alpha carbon. But we also have beta amino acid. So we can see the structure. It must be the amine group connected to the beta carbon. And for different side chains, we have different amino acid. If the side chain is hydrogen, we got glycine. And if it is a phenyl group, then we got a phenylalanine here. So the different amino acids, we have 20 different kinds of amino acids. We can use these 20 different kinds of amino acids to make different variety of peptide chains. So between the each amino acid, how did they connect it to each other? They are joined by a peptide bond. It's a covalent bond, a peptide bond. And the reaction is reacted by a condensation reaction. And the molecule of water is removed, and then we got a peptide bond. And on the contrary, if we hydrolyze this peptide bond, then we can get two molecules of amino acids. It's happened on the hydroxy group and the acid group, the condensation and hydrolysis reaction. We can say amino acids is a really basic block of the peptide chains and also the proteins. We can use different amino acids to make peptides, and then we can assemble the peptides into a big, larger protein. And we have mentioned the 20 different amino acids in polypeptides synthesized on ribosome. But do we need to learn all of the 20 amino acids? Actually, we can classify them into different groups. Like we can classify them by essential or non-essential or conditionally essential. Essential means the amino acids must consumed in the diet, and non-essential can be synthesized in the body by ourselves. And conditionally essential means they cannot be synthesized due to the illness or lack of necessary precursors. Like in the infants, some of the infants, they don't have enough precursors or the enzymes to make some amino acids, so they must be consumed in the diet. Well, let's see the structure of the common structure features of these amino acids. Because the active site of enzyme are asymmetric, so it's causing the reaction of the enzymes they catalyze to be stereospecific. So here what's in our body, they are all alpha amino acids. So if we have some beta amino acids, the enzyme could not conduct the reaction. And also because the alpha carbon atom make its chiral center, so we can get some isomers of different kind of amino acids. Like the stereoisomers is in our human bodies. And then if we use D amino acid residues, some of them are caused by our human body's enzymes to transfer the alpha amino acids into the D amino acids. And cells are able to specifically synthesize the L isomers of amino acids. And we can also classify the amino acids by its R group, the different properties of its R group. Like we can classify them into nonpolar and polar groups. And nonpolar group, we can classify them into aliphatic R groups like alanine, leucine, isoleucine. They always have hydrophobic interactions because the aliphatic R group. And the aromatic R groups like phenylalanine, tyrosine, and tryptophan, and glycine and proline, they are all nonpolar amino acids. And glycine and proline are a little bit special because glycine is the simplest structure. The R group of glycine is only a hydrogen, and proline is also special because it's a cyclic structure. We can here is the proline. The amine group is connected to the alpha carbon, so it's made of 5-atom ring here. It's proline. So we have nonpolar amino acids. We must have polar amino acids, like the uncharged R group serine, tyrosine. They always have some hydroxy group to make its uncharged R groups. But some of them are positively charged, and some of them are negatively charged. Like the asparagine and glutamine, they are the amides of aspartate and glutamate, respectively, and they are easily hydrolyzed by acid or base. Histidine has an ionizable side chain with a pK near neutral. So histidine residue facilitates many enzyme catalyzed reaction by serving as a protein donor or acceptor. Cysteine is also special because it can form a covalent bond, which is called disulfide bond, to make the two cysteine into a joined by a disulfide bond. We can see later the bait sheet structure peptides are always connected by this disulfide bond. Besides the 20 common amino acids, we also have uncommon amino acids. For now, there are over 300 different kinds of uncommon amino acids has been discovered. Well, they must have their meanings in our human bodies because like they can alter the protein's function or increase or decrease the protein's activity. That's the classification of amino acids. For the peptides, we always classify them by the length of the peptides. Like the peptides less than 10, always we call them oligopeptide. They consist of a chain of only a few amino acids, whereas the polypeptides usually consist of a longer chain of amino acids, usually have molecular weight of below 10,000. Well, if it's above 10,000, we always call it a protein. And the oligopeptide, we can also name them like this. Here, there is an example. There are 5 amino acids to form a peptide chain. So we call it a pentapeptide because there are 5 amino acid residues. They form 4 peptide bonds. So if we have only 1 amino acid, it's not, because it's not a peptide, it's only amino acids. So there's no other name. But if we have 2 amino acids to form a peptide bond, then we can call it a dipeptide. If we have 3, that's a tripeptide. And if we have 5, that's a pentapeptide. And 6 is a hexapeptide. That's how we call the peptides. That's some structural knowledge about the amino acids and peptides. Here we can see another video to more specifically introduce the central dogma of the genetics, the biosynthesis of peptide. And we can see the differences between the
We
biosynthesis.
can't see your presentation. Please show your screen.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, that's the biosynthesis of peptide chain. You can see it start from the N terminal, then N terminal of the peptide to the C terminal, the carboxyl terminal of the peptide. But for the solid phase peptide synthesis, is mostly commonly start at the C terminal of the peptide, and then proceeds towards the amino terminal, the N terminal of the peptide. So the protein biosynthesis in living organisms occurs in the opposite direction. So let's see the chemically synthesis of the peptide chains. We always use solid phase peptide synthesis, which is also called SPPS. Usually we use FMOC chemistry. We use this protecting group, FMOC, as a protecting group. It could protect the amine group. Here is a mechanism of the deprotection procedures. Here the FMOC is protecting the amine group here. And then we use a base like piperidine to take out of protein, hydrogen. So the electrons transferred, and then this carbon and nitrogen bond was cleaved, and the free amine group is available for the next reaction. And this is called a deprotection. And the FMOC is called a protecting group. So for the coupling procedure, we have a resin, and here is a free amine group. Then we use the first amino acids with the protecting group and do the deprotection, and we got now here a peptide bond, and here another free amine group. Then we do the coupling again. We use the second amino acid, and now we have another peptide bond here, and here is with a protecting group, and we do the deprotection and coupling, and later we have longer peptide chains. And for the final step, we can cleave the resin down from the peptide chain. There is a cleavage step. Then we can have a peptide chain by this solid phase peptide synthesis. Because the resin is a solid phase, it cannot dissolve in water. So all the reactions happened on the resin. It's called solid phase, SPPS. Okay, that's the basic knowledge about the amino acid and the peptide. Then I can show you some about the secondary structures. Because of the time, I talk too slow above, so I may pass some of the pages. Well, the sequences, we just introduced the amino acid and formed the sequence, and it's called a primary structure. It's actually the sequence of the peptide. And then the primary structure could form a secondary structure, like alpha helix or beta sheet structure. And then the structure can assemble with longer and larger peptide. They can have tertiary structures, and different subunits could assemble together to form quaternary structures, a larger protein. For us, for now we can only study the primary structure and secondary structure. But the second structure is a stable arrangement of amino acid residue, giving rise to recurring structural patterns. The secondary structure. Here is a structure of a host-defined peptide. I download this model from the protein data bank, PDB. And this image shown here was made by using the data from the entry with PDB ID 2L24. And later, some of the models are all from this host-defined peptide. Well, we can study some of the important features of protein structures. But the function of a protein depends on its structure, now we know. And the most important force stabilizing the specific structures maintained by a given protein are non-covalent. But the protein structures are not static. And the spatial arrangement of the atoms in a protein is called its conformation. And proteins in any of their functional folded conformations are called native protein. The protein's conformation is stabilized largely by weak interaction, like the hydrogen bonds, hydrophobic effect, ionic interactions. Here is the covalent bond. We already introduced the disulfide bond between two cysteines, like in the insulin. In the insulin, there are several cysteines, and they can form 3 disulfide bonds. One is within the chain A, in a peptide chain, and the other 2 are between the 2 chains. So they form 3 disulfide bonds, and these disulfide bonds could stabilize the protein or peptide structures. And free Gibbs energy is also very strong. So that's the non-covalent bond. But you see the other hydrogen bond, ionic bond, dipolar interaction, hydrophobic interaction, they are very weak interactions. But covalent bond is a strong interaction. Hydrogen bond. A hydrogen bond is partially electrostatic attraction between a hydrogen atom, which is bound to a more electronegative atom or group, such as nitrogen, oxygen, or sometimes fluorine. The hydrogen bond donor and another adjacent atom bearing a lone pair of the electrons is called the hydrogen bond acceptor. Actually, hydrogen bond it's very important in human macromolecules. Proteins are nucleotides containing many nitrogen hydrogen bond and oxygen hydrogen bonds. So many drugs molecules contain electronegatively atoms that can form hydrogen bonds. And salt bridge. Salt bridge is also called the ionic bonding. We can think about the salt, the sodium chloride, for better understanding of the salt bridge. The sodium and the chloride, they can form an ionic bond. So does the positively charged amino acid and the negatively charged amino acid, like the lysine. Lysine is positively charged, and the glutamic acid is negatively charged. So there is a electro interaction, which is ionic bonding between the two amino acids. It also can stabilize the protein structure. Here is a model of what we study now. It's a peptide and a protein here. The green one is a protein. Here the inhibitor, the green one is a peptide inhibitor. And we can see there are many short bridges between the peptide inhibitor and the protein, the enzyme. And the other one is van der Waals force. It's a dipole-dipole interaction involving the permanent electron dipoles in groups such as carbonyls. Transient dipoles derived from fluctuations of the electron cloud surrounding any atom, and the dipoles induced by interaction of one atom with another that has a permanent or transient dipole. And these van der Waals interactions are substantial only in well-packed protein, or in an interaction between a protein and another protein, or other molecules at the complementary surface, van der Waals force. And also hydrophobic interaction. We can see here if X and Y, they are all hydrophobic. Both of them are hydrophobic molecules. So they can not form the hydrogen bonds with the solvent, the water. So X and Y, they have a hydrophobic interaction. They have a tend to cluster together and to get away from the water. So actually there is a force between them that's called hydrophobic force, hydrophobic interaction. For some of the amino acids in our peptide chains, they are always hydrophobic, like the leucine, isoleucine, valine, phenylalanine, and tryptophan. They can form a hydrophobic surface, or for bigger proteins, they can form a hydrophobic curve to induce some reactions or to show some activity of them. The predominant protein secondary structures are alpha helix and beta sheet, and some of them are beta tens. If the secondary structure are not defined, they can also called a random coil. So we see the alpha helix structure. Alpha helix structure is a very common form of the secondary structure, and the right-handed alpha helix is a common form. So if we have a right-handed alpha helix, we also have a left-handed helix, but left-handed helix is not a common one. About 1/4 of all amino acid residues in proteins are found in alpha helices. Some of the amino acids could affect the stability of alpha helix, like the we mentioned the two very different amino acids, glycine and proline. They are quite different because they have the least porosity to form alpha helices. In proline, it's easier because the nitrogen atom is part of a rigid 5-atom ring, and the rotation about alpha carbon and nitrogen bond is not possible. So proline residue introduces a destabilizing kink in alpha helix, and glycine has more conformational flexibility than other amino acid residues. So always glycine is a kink in an alpha helix. Some constraints that affect the stability of an alpha helix: intrinsic propensity, the interaction between these R groups and the balconies of adjacent R group, and the occurrence of proline and glycine residues, and the interactions between amino acid residues at the end of helical segment and the electric dipole inherent to the alpha helix. And so there are also some beta conformations. You can pass here, you can learn it later by yourself. Beta sheet structures. Two different kinds, anti-parallel beta sheet and the parallel beta sheet. You can see that's because of the nitrogen bond to make different anti-parallel or parallel beta sheet. Later is our case study. Here is a paper that we published last year. It's a very typical paper about the antimicrobial peptide research, from the discovery to the identification to the function screening, and then the mechanism study, and the in vitro and in vivo pharmacology study. So I can use this paper to give a brief introduction about this research area. It's called identification of new dermoseptins. It's a name of a family of antimicrobial peptides, but it's a little bit interesting because it has a self-assembly tendency, and its function is membrane disruption, biofilm eradication, and the infected wound healing efficacy. We introduced the mechanism. Is discovered by Zaslov and his colleagues. So we know the host defense peptide could be discovered in the amphibians. So this peptide is also found in a frog. It's called Philomedusa salvagei. It's from South Africa, the Amazon forest, I think. So this frog, we breed them in our lab, and then we get its frog skin secretion. And then we use the frog skin secretion to do the molecular cloning, because the frog skin secretion, there are mRNAs inside of the secretion. So we can reverse transcript the mRNA, and then we can get the DNA sequence of the precursor. And then we can have the DNA information of these different peptides. And we can use the DNA information to predict the structure of the peptide structure, right? But it's only a prediction. So we need to do them parallel, like one is molecular cloning and the other is to use HPLC mass to identify the peptides, the mature peptide in the frog skin secretion, to make sure it's established in this frog skin secretion. So we use a complex chromatogram in 240 minutes, provided by the crude skin secretion analysis performed by reverse phased HPLC, and following the MS fragmentation was used, and this was detected by MS, and we found in this elutes about 117 minutes, the elute sample, we can detect the existence of this mature peptide, and therefore the truncated isoforms of the peptide were identified by MS analysis in primary structure, and the peptide was also validated. Structure of this one natural peptide was chemically synthesized later, and they were confirmed to have the purity of 95%. So we later used the chemically synthesized peptides to do the downstream experiments. Well, in addition to the molecular simulation, we also use CD. This is a molecular simulation. We can see it's an alpha helical structure. And in addition, we use CD here to further experimentally determine the secondary structure of this peptide. And we found the peptide existed as a random coil in an aqueous solution. Here, the green one. This way means the peptides mainly adopt a random coil structure. But in a membrane mimic environment, like in the red one, we use 50% TFE. It's a membrane mimic environment. We got a very typical alpha helical structure graph here. So we can use experiment to further determine the secondary structure of this peptide. And this means this peptide, dermacebtin, was obviously induced to a typical alpha helical conformation by the membrane mimic environment. And also, actually, we also do other studies, like we can also see the alpha helical structure can be induced by the bacteria. And then they do the function screening, and we see this family, peptide family, is always antimicrobial. So we test the antimicrobial activities. So we use a minimal inhibition concentration, minimal bactericidal concentration, and the minimal biofilm inhibition concentration, and the minimal biofilm eradication concentration. Use these 4 assays to test the antimicrobial activity of this peptide. And we found the MIC, MBC, MBIC, and BEC value of this peptide to Staphylococcus aureus and to E. coli, and even to Pseudomonas is very good. It's very good. It's even less than 8 microgram per mil. So we already make sure the antimicrobial activity of this peptide. So the next step is we need to investigate the mechanism of this peptide, the mode of action of this peptide. And here is the mechanism study. Well, the peptide localization experiment was performed by observing FITC labeled peptide under the sub-MIC value, because the MIC value could inhibit or kill the bacteria. So we use the sub-MIC value. And using the confocal microscopy, and all the bacterial cells could be stained with green color by a dye, which is called cytonine, while the bacteria with disrupt cell membrane showed red color by staining with the PI, Propydium, the PI, another red color dye. And comparing to the control, the permeability of the cell membrane of the bacteria, the MRSA, was significantly affected by the treatment of this peptide. Besides, the bacterial cell displayed permeabilized membrane at MBC concentration, since most of the bacteria was stained with the red color. Furthermore, a scanning electroscopy (SEM) was performed to investigate the effect of this peptide on the cell membrane. And the control was observed with very smooth and intact surface. You see here, very smooth and intact surface for the Staphylococcus, and this is for the Pseudomonas aeruginosa. But that's a control group. But the normal cells were evidently lysed, followed by fusing, clumping, and disintegration by treating with our peptide for an hour. Here, an abundant cell cluster were evidently observed in the environment. Additionally, this is Pseudomonas aeruginosa, incubated with this peptide, also displayed aggregation, shrinking, and collapse, both of the planktonic bacterial cells and the adherent cells embedded in the biofilm had been damaged in the presence of this peptide. It's very clear evidence. And additionally, it was found that the dermacectin, the peptide molecules, was spread on the surface of the gram-negative bacterial membrane, adopting alpha helical conformation, and this is verified by based on the molecular docking study. Like I said in the title, there is a little bit self-assemble activity, so we also test the self-assemble activity and the property of the peptide by using molecular cloning and DRS, and also another microscopy to make sure this peptide is really have a self-assemble tendency. This is a atom force microscopy, and this is actually the self-assembled particle of this antimicrobial peptide. And its effect on the cells, and we already know the mechanism, so then we can try if this peptide could be used on the animal models. So we use a MRSA in a skin infection model to see if our peptide could treat the infected wound. We can see here the efficacy of this peptide on infected skin wound healing. Here we use marine skin and infected by MRSA infection. And the treatment of this infection model was assessed by the validation of proliferation and stable bacterial viable counts. And this peptide potently inhibited the MRSA growth in the infected urine skin, and the variable count result of this peptide treatment group displayed a significantly decreased compared to the saline control group. And after a third day treatment, the variable count remained at a low level, and the wound surface started to scab with shrinking. And compared to the control group that failed to control the growth of the MRSA, and the peptide treated infected wound was efficiently to decrease the bacterial variable count and showed a very distinct wound healing. This peptide treated wound initiated to scab 3 days post-infection, while the control group appeared to scab 7 days post-infection. Moreover, we can see the ascarotic exfoliation gradually appeared after 7 days medication. That's a peptide treated group, and that's a control group. So we can give you a brief summarize. The antimicrobial peptides from the skin secretion of this frog, Philomedusa, has been identified in this study, and the cationic peptide dermacectin exhibited a significant broad spectrum antimicrobial activity, and and also the planktonic and the biofilm of persistent infections associated with the weak toxicity for mammalian cells, and the marine membrane permeabilizing ability of this peptide was confirmed. And importantly, it demonstrated potent efficacy of the treatment of MRSA-infected murine skin model. And furthermore, what beyond our expectation is that this peptide displayed a self-aggregation property, which also makes it a promising suggestion for the infection-proof biomaterial development. So that's the case study of this host-defense peptide investigation. Okay, the last part is peptide bioinformatics. I can give you the resources, and you can. It's very initial knowledge, very basic knowledge. So you can use these resources to have a self-study. The first one is NCBI. NCBI. The website is here, and if you click and you can find the BLAST on the right panel. BLAST. And what is BLAST? It is the short for Basic Local Alignment Search Tool. And what we use usually is nucleotide BLAST. And for this protein study, we use protein BLAST. And if you click the protein BLAST, here is the interface. And you can enter the sequence. Actually, you can just input the sequence with like this, or very simple sequences, and then you do the BLAST, and you can find some information about this sequence, the one that you want to find. Like you can see here, the description of your sequence and the graphic summary and the alignment and taxonomy of your sequence. If you click the description, you can see many different sequences. And here on the right side panel, you can see the e-value here. If the e-value is small, it means there is more identity of your peptide sequence and the peptide sequence from the database. So you can see here the identity is 100%. So that means your peptide sequence is identical to this peptide sequence here in the database. Actually, it is this sequence 2L24. And for the graphic summary, you can see the color. Color means the alignment scores. And for the alignment panel, you can check many other informations, like your queried sequences and your subject sequences. And here you can see the structure, related information. You can see the structure here. And then the structure of the peptide we input to R24. That's the secondary structure. Also, you can click here to R24 to see more information. It's the chain A of hemagglutinin. And the keyword, the source, the organism, the reference, the authors, the title of the paper, and the journal. You can also check here. It's from the Journal of Medicinal Chemistry, JMC. Another useful resource is PDB, Protein Data Bank. You can search your protein here. And if you get a new protein structure, you can also deposit your structure into the PDB. You can use your sequence or the PDB ID search in this box. And then you get these information, structure, summary, 3D view, sequence, similarity, and experiment. And here, display file and download file, you can download a FASTA sequence. FASTA sequence is very useful and very easy to use. It's the very commonly used in the protein study. And the PDB format is also very important because you can use your PDB format and use specific software to display the sequence, the structure in your computer. Like here, if you download the PDB file, you can use the PyMo. PyMo is some of them are free download for the education institutions. PDB, and if you open it by PyMo, you can get this structure. This structure, that's a 3D structure. But if you want to do some modifications, you can have a modification to make it more beautiful, or you can see it more clear. Like you can use the stick mode or the surface, or you can drag it to different angles, and cartoon and cartoon-based side chain and main chain, and label the amino acid residues and use the rainbow colors. And you can show your talent to make a beautiful picture. And for this one is made by the PyMo and the protein structure from the PDB data bank. 1224. You can also, if you cannot install the PyMo software, you can also use it online. In the PDB here, you can see cartoon or backbone or surface, different kind of styles of showing the 3D structure. Here is another one. This is for the prediction of the secondary structure. You can see that here is it's a predicted. You can use this software online web server to have a prediction. And this is also a prediction web server. Can predict the physiochemical properties and polar residues and non-polar residues here, statistics. And another larger website is this one, EXPASY. It's a database of different kind of web servers, but all about bioinformatics. It's a resource portal, so you can go to this website and find more web servers that you want to use, like the proton para is used for prediction of the property of your protein or peptide. And this is called Rampage to use to identify if your molecular simulated peptide or protein structure is available or not by the Ramachandran plot. Here is the Ramachandran plot. If you want to study more, you can start from the Ramachandran plot. It's about the phi and the psi angle. Okay, that's all about our lecture today. We talked about the host-defined peptide and some basic knowledge about amino acid and host-defined peptide and the secondary structures and the biosynthesis and the chemical synthesis of the peptides, and the case study about our host-defined peptide investigation, and then some useful bioinformatic web servers or tools was also introduced to you. So you can have a further study about this field if you like. And that's all for today's lecture. Thank you. Hope to see you guys again.
Thank you, doctor.